Inside the ovary of a cherry flower, a mass forms on the inside of the ovary wall. This is the start of an ovule. As the cell mass grows, a megaspore mother cell forms within it, and one or two protective integuments form around it. The megaspore mother cell undergoes meiosis to produce 4 haploid megaspores. 3 of the megaspores disintegrate. The remaining megaspore undergoes mitosis without cytoplasmic division to produce a cell with 2 haploid nuclei. 2 more rounds of mitosis without cytoplasmic division yield a cell with 8 haploid nuclei. Each nucleus migrates to a specific position and the cytoplasm divides. The result is a 7-celled embryo sac. This is the mature female gametophyte. 6 of the cells have a single nucleus. One of the cells in the embryo sac, the egg, is located directly behind a narrow opening in the integuments called the micropyle. On either side of the egg are synergids. The one large central cell contains 2 haploid polar nuclei. Inside the anthers are 4 microsporangia, also known as pollen sacs or pollen chambers. Each pollen sac is filled with diploid microspore mother cells. These diploid cells undergo meiosis and cytoplasmic division to produce haploid microspores. The microspores separate from one another. Each develops a thick wall around itself, and each haploid nucleus undergoes a mitotic division. The final product is a haploid pollen grain, which is the male gametophyte of flowering plants. The pollen grain contains 2 haploid nuclei. One is the generative nucleus, the other is the tube nucleus. The generative nucleus will divide by mitosis to form 2 sperm nuclei. The tube nucleus will form the pollen tube that will grow down the style towards the ovule. The stage has now been set for pollination. Drag a pollen grain to the receptive stigma to see what happens next. When a pollen grain lands on a receptive stigma, it germinates. The tube nucleus forms the pollen tube, which grows down the style into the ovary. The generative nucleus divides by mitosis to form 2 sperm nuclei. The pollen tube will carry the sperm nuclei up through the micropyle to the ovule, where double fertilization will take place. The first sperm nucleus fuses with the egg to make the zygote. The second sperm nucleus unites with the 2 polar nuclei to make the triploid endosperm. Once double fertilization has occurred, the ovule begins to mature. The zygote will divide by mitosis to form the embryo, while the integument hardens to form the protective seed coat. The final result of double fertilization is a seed consisting of a diploid seed coat, a triploid endosperm, and a diploid embryo.